Hello, my name is Dr. Natalie Fey and I'm a senior lecturer at the University of Bristol working in the School of Chemistry. I'm going to give you a relatively short presentation on how you can use some computational chemistry to map out chemical space, and because it's January 2021 I can't come into work to do this, so you get to see my home office as the background a little bit as well. But I think that's quite enough of seeing me, and it makes the files really big, so I'm actually going to turn off the camera for the rest of these recordings. So as I mentioned before, I'm a computational chemist. So rather than the groundbreaking Lego figure that you're seeing at the moment with the blue nitrile gloves working in a laboratory environment, all of my work is actually undertaken on a computer. And I'm planning to tell you a little bit about what that means and what sorts of results you can expect from this sort of work. So before I tell you more about my work, let me start out by just talking a little bit of scientific computing. And I obviously have an ulterior motive here because I'm the program director for our new degrees in chemistry with scientific computing. And they're basically about all of the areas where computers make it easier and better to study chemistry and molecular structures. So if we start here at the top, let me just change my pointer so you can see what I'm talking about. So what I've put up here is essentially a treasure map of molecular structures, and in my talk today I'll introduce you a little bit more into how we can use computational approaches to map out this chemical space. And then if we go clockwise around this cycle, the next bit we get to is automation. So what we've got here in the middle is the core of an automated chemical synthesis platform. And what it does is it has these robotic arms to portion out chemicals and to take samples from up to 48 reaction vials. So what you can do, it's also a glove box, is you can control the reaction conditions and essentially carry out 48 reactions under controlled conditions rather than just one or two. Down here we've got a series of NMR samples, and basically our spectrometers can run all night with a robot just feeding in these sample tubes, and all of the collections are automatic. We can also use computers to control other instrumentation. So this is essentially the core of an X-ray diffractometer. And again, all of the setup and all of the data collection here are controlled by a computer, and they can also be automated. And then over here, we've got the inside workings of a laser spectrometer. And again, it doesn't rely on people firing the lasers anymore. This is now all controlled by computers. Moving on then, we are looking at the underpinning chemical theory, and what I've just put up is the so-called Schrödinger equation, and in its full form it allows us to model all atomic and molecular structures, but it becomes fiendishly complicated. So we're actually using computer models to come up with the best way of making sensible approximations to the data, and also to compute elements of it. And then finally we've got simulation here, and what I've put up is firstly an energy profile, and then also the active site in a metalloprotein. So with simulations we're able to map out the course of reactions and also to analyze the structural changes and how these reactions might actually work. So from my point of view, the really big idea in chemistry is molecular structure, because all of chemistry is about observing, analysing, making and changing molecules. And let me just give you an example. So what we've got here is cyclohexane, and if you burn this in air, you can find out relatively easily that it has a molecular formula of C6H12. And then you start thinking about how could 6 carbon atoms and 12 hydrogen atoms be put together. And with a strong cup of tea and a little bit of thinking, you might figure out that because carbon makes 4 connections and hydrogens just make 1, you should draw a cyclic structure consisting of 6 carbon atoms. And that then gives each carbon atom 2 connections, and then you satisfy the other two connections by putting the hydrogen atoms there. And if you think about it, in a lot of the chemistry we do, we actually need to communicate about these molecular structures. And it would become really, really dull if we were sitting there and just writing out all of these atom types explicitly. So just sitting there writing carbon, 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 hydrogen, 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 hydrogen. So chemists generally use a shorthand, and formally that's called a skeletal structure. So what we do is, because so much of chemistry is about carbon and hydrogen, we've built up shortcuts around them. So in skeletal structures, we represent the carbon atoms by corners, and we try and get the geometry roughly right as well. And then hydrogens are not generally shown unless they're important to reactions. So in this particular case, we're not looking at the reactivity, so we're just not drawing them in. And these structures are three-dimensional, so if you were actually looking at this from the side, rather than being a completely flat six-membered ring, it's what's called a chair conformation. And this is an attempt to draw this a little bit as a 3D effect. So in computational chemistry, we have lots of different ways of representing these molecular structures, and I've shown you 3 examples here. So if we start over on this side, this is, for obvious reasons, called a skeletal or stick structure. So the bonds are represented through sticks, and you can maybe just about make out that the colors also change. And if again we look at this from the side, then you're perhaps starting to appreciate that these three-dimensional structures are actually quite complex, and also that this wouldn't be a terribly comfortable chair to be sitting on. In the center, we've got a ball and stick structure, so the atoms are now represented by spheres, and they're scaled spheres, so the hydrogens are much, much smaller than the carbons. And again, you can see a little bit of the three-dimensional structure if you look at it from the top, but you can start to appreciate a lot more if you look at it side on. And then over on this side here, we've got what's called a space-fill model, nothing like an obvious name. And if you could reduce yourself to the size of atoms, which is not possible, then this is probably closest to what you would see. So atoms form bonds by sharing electron density, and that's represented here. And then you can also see the relationship between the sizes of the larger, darker grey carbon atoms and the smaller, lighter grey hydrogen atoms. And if you look at this one side on, it's starting to look a little bit more comfortable as a chair, but I still wouldn't really want to spend a lot of time sitting on it. So how do we actually know about molecular structures? Well, there are a range of techniques, and I've mentioned some of those already. So you can use spectroscopy, and there are different flavors of spectroscopy depending on the wavelength of the electromagnetic radiation you're using. So you can have infrared spectroscopy, UV visible spectroscopy, which covers the visible range of light, and then nuclear magnetic resonance spectroscopy, and also laser spectroscopy. You can also use diffraction techniques, and I've mentioned X-ray crystallography, where again you use radiation, but this time it's diffracted by the atoms in molecular structures, and you can solve that to study structures. And then you can use computation, and you can use computation both to interpret experimental data, but also to make predictions about compounds you may not have made, or ideas about the mechanism of a reaction. So I'm a computational chemist, and these are 3 examples of computing clusters we have used in the past. So this is a Beowulf cluster just set up of these individual units and lots of networking cables, and they're really difficult to keep cold. So people have then moved on to rack-mounted computing clusters, and again you can see lots and lots of cables there. And nowadays we have high performance computer, and this is a shiny image of the university's high performance computing cluster. It's called called blue crystal, and there are essentially lots and lots of these rack mounts behind these in these cabinets, all connected up and networked together. So for about 5 minutes it was one of the fastest university hosted computing clusters, but then someone else bought a faster machine, so they get trumped very, very quickly, but it's still a very, very impressive computing cluster. Right, so here is our warning, and I actually bought that calendar just for this image, and it's that computers let you make more mistakes faster than any invention in human history, with the possible exception of tequila. So let's see what we've done about this in my group. To illustrate this idea of using molecular structure to say something about the properties of molecules, I wanted to start out by telling you a little story about a molecule present in chili. It's called capsaicin, and it's the name is derived from the botanical name for chili, which is capsicum. A lot of the pictures I'm using and the ideas are actually covered in the Picture It Chemistry blog, and I've put a link to the blog post at the bottom of this slide if you're interested. So chilies are quite versatile, and they're used in a number of different ways. The one you're probably most familiar with is to spice up the food for mammals, and it's important to recognize that this only works in mammals. So apparently birds can't detect the heat of a chili, and it's thought that this arises because they don't have that receptor for chili heat. Chilies are also high in vitamins and various trace elements, and it's thought that what we know as the chili heat actually evolved to keep away those greedy mammals from picking the fruit and not distributing them widely. It can be used as a food preservative, but also as an elephant repellent, a bear spray, and a pepper spray. And finally, people noticed that it can give pain relief, and that's again thought to be related to the capsaicin sitting in the vanillin receptor, which is not just related to scent, but potentially also to pain. The molecules found in chilies are together called the capsicinoids. And the effect of heat and spice is considered to arise because they dock into a enzymatic structure that is actually the vanillin receptor. So what we can do is we can compare the structure of vanillin with a range of different molecules. So for capsaicin, you can see again there's the same head group, and a little bit further removed there is also a carbonyl group, and then further along the chain we've got this double bond. In dihydrocapsaicin, we've essentially hydrogenated that double bond, so this is now a single bond, and then in nor-dihydrocapsaicin the chain is actually one carbon molecule shorter. Another smelly molecule, so vanillin, as the name suggests, smells of vanilla, but another smelly molecule is benzaldehyde, and benzaldehyde smells of bitter almond oil. And again, it has the aromatic ring and this carbonyl group in common with both vanillin and the capsaicins. So, as I said before, we share the aromatics, they're present in all of them. The hydroxy group is present in all of the capsaicins. The methoxy group is also present in all of these. And then the formyl group is unique to vanillin, also present in benzaldehyde. And then in the capsaicins, that carbonyl is present, but it's slightly further removed from the aromatic. It's quite a flexible molecule, so it can actually adjust its shape to still fit into the vanillin receptor in the bodies of mammals. Just to fill that in, this is then called an amide group because you've got an NH group adjacent to the carbon. So one of the reasons molecular structure is so important is that it often allows you to predict some of the properties of molecules. And one of those properties is solubility. So one of the rules of thumb all chemists use is that like dissolves in like. And if we look at the structure of vanillin, then it has this hydroxy group, and it looks a little bit like one side of a water molecule. But it also looks quite a lot like an alcohol molecule. This is ethanol. So again, it has this hydroxy group and then a slightly longer tail of carbon atoms. So the solubility of vanillin in water is quite low. It's only 0.07 mole per decimeter cube, whereas it goes up in ethanol, and that will be because ethanol has this longer tail. If we look at capsaicin, again, we've got the hydroxy group, but we now have a much longer tail of carbon atoms. And what we find with capsaicin is that the solubility in water is now very low. And it is a bit higher in ethanol, but it's still quite low. If you then look at this tail, what we normally say is when we've got long chains of carbons and hydrogens, they're called lipophilic, so they're quite greasy tails, and that comes because if you look at the structure of a lipid, you've got this lipid head group here and then these very long tails. And again, under the concept of like dissolves in like, we can then predict that capsaicin should actually dissolve relatively well in oil, and it does. So one of the reasons why a lot of spicy Indian food is served with lassis, so yogurt-based drinks, is because it dissolves the capsaicins quite well and it takes the heat of that chili away. And we can now relate this to the molecular structure, so that's what's going on. There's also quite an interesting study where people were interested in using capsicinoids, so molecules related to the capsaicins, but not making them sting quite so much. And all they did chemically was replace this NH group with an ester group, and they found that it still blocks the vanillin receptor, but it isn't detected as this hot and burning heat. So just making a small modification to the molecular structure can also change these properties. So why am I talking about all of this? Well, as I said before, I'm a computational chemist, and we can use molecular structures to predict these properties of molecules. In my research, we're interested in determining the properties of ligands. And by ligands here, I mean small molecules that combine to transition metal complexes. And these transition metal complexes are often used in organometallic catalysis. So being able to understand and predict the properties of different ligands allows us then to control the chemical reactions. You can characterize the properties of ligands. So if we look at this example over here, this is quite a large ligand and it has some unfavorable steric interactions with the metal complex on top. There are also some calculated structures here for different types of ligands. So as you can see, this one has a red area, which means it's a good electron donor, and then these more blue areas, whereas this type of ligand is much more electron withdrawing, and there isn't quite as much electron density available, so it's less red. What you can then also do is you can use these properties to map out how the ligands relate to each other, so whether they're more similar to each other or more different from each other, and from that you can then derive what are called structure property relationships, so predictions about how these ligands will react when you actually use them in organometallic catalysis, for example. So what we've done here is we've calculated the properties of this same ligand in lots and lots of different coordination environments. And then we've used a statistical mapping technique, and it's called principal component analysis, to determine which ligands are similar to each other and which ones are more different from each other. And I don't really want to go into the details of this technique at the moment, but each of these spots corresponds to a single ligand. and they are color coded here by their chemical identity. So R corresponds to alkyl groups, AR corresponds to aryl groups, and then different substituents are different shapes and different colors. And what you can hopefully see is that the ones that are coded to be similar in terms of their chemistry are quite close together, and the ones that are different in terms of their chemistry are quite a long way apart. So that's why I keep talking about these maps. So what you can then do is you can go in and you can say, okay, this system here called tri-tertiary butyl phosphine is quite interesting. Which of these systems are nearby, and can I use them to do the chemical reaction better? You can also zoom in on this, and here we've got a problem where we've got a system here called triphenylphosphine, and it's okay for catalysis, but it's not great. On the other hand, these types of systems where we've essentially inserted an oxygen between the phosphorus and the phenyl group make much better catalysts. They're quite quick, but they're not very stable. So what my colleague Paul Pringle wanted to do was to design systems that react with the speed of these systems. They're called phosphides, but are much more stable. And he came up with different designs. So the first one was to replace one of the phenyl groups with a fluorine, and that moves us over here on our map of chemical space. And he also likes this cage-like system here. It's an adamantyl phosphine. And again, he was interested in predicting what would happen when we replace a phenyl group with a fluorine. And as you might expect, it moves the system over here. And then he also likes these weird ring systems. And again, by putting a fluorine group, this puts us over on this side of our map. And this makes really promising systems. So on the strength of looking at them on our map, Paul's student then actually prepared these ligands and they tested them in catalysis, and they were actually pretty good systems. So this is just an example of how powerful it can be to look at molecular structures and use these to predict the properties. So there's an element of assembling Legos in predicting molecular properties, because what you can do in the computer is to combine the different substituents, the different donor atoms with each other, calculate the properties, and so predict whether they are going to give you the behavior you want, or whether a different modification would be more useful. And that's essentially what we did in the project I've just talked about. So here is another example, and rather than looking at the monodentate type ligands, which we've looked at so far, where there's a single phosphorus atom, we were interested in the properties of so-called bidentate ligands. So what you've got here is two phosphorus atoms. Each of them can coordinate a metal center. They're connected by a molecular fragment which we call the backbone, and you also have substituents on each of those. And I've turned it into a bit of a cartoon here just to highlight that point. So computationally, you can change the backbone, you could change the donors, although we didn't do that in this case, and you could also change these substituents, and you can do so systematically. So this is again one of these maps of chemical space that comes out if you calculate lots of properties, process them with statistical approaches, and then look at which ones are similar to each other and which ones are different from each other. And here they're color coded by substituents, and you can again see that they arrange themselves in stripes, with the spots that are close to each other relating to ligands that are much more similar, and those that are further apart being very different from each other. that sort of idea got picked up from a review article I wrote and featured in Chemistry World. And Chemistry World is the members magazine of the Royal Society of Chemistry. And they were really interested in how you could use this idea of computationally assembling molecular structures to find new types of molecules, new types of functionality. So this is the cover picture that they used for this. So to bring these ideas together, what I've hopefully showed you is that we can do calculations on molecules, on transition metal complexes, which are catalysts, that are relevant to synthesis, that are the types of molecules that synthetic chemists can make and use. And with computational chemistry, we're not restricted to just the molecules that have already been made, but we can also consider things that are novel or unstable or transient. So it gives us a much broader opportunity to look at the chemistry involved. In terms of synthetic chemistry, I've mentioned the spectroscopies a little bit, and also X-ray diffraction. So they can all be used to detect and analyze stable species and relate those to the properties we observe. And we can also use we can use synthesis to optimize reactions, so to find the best reaction conditions, and that's something that's really, really challenging computationally. So overall, they really are better together. You can't do synthetic chemistry nowadays without having the computational predictions and using computers to control your instruments and do the data analysis. But it would also be pointless to try and make predictions without ever tensioning them against the experimental data. So if you remember nothing else, then it's certainly worth bearing in mind that these techniques are best used together rather than in isolation. So that really just leaves me to acknowledge some of the people who've done this work and who've helped with this work. So first of all, the Picture It Chemistry blog, which I mentioned, my co-editor initially was Jenny Slaughter, and this has now been taken over by Chris Adams. Claire McMullen, Jesus Robert and Derek Durand have done some of the work I've talked about. And I also wanted to thank my group, both past and present, for all the contributions they've made. I have a number of academic colleagues and collaborators, and they're named here. And we're based in the Bristol Centre for Computational Chemistry, which is a really nice environment to do these calculations in. And then finally, all of these people have contributed some of the funding to do this work. And with that, I really just wanted to thank you for listening to this. And I hope you've learned some useful things about how you can use computational chemistry to map chemical space.